Thanks. Good morning. Good afternoon, actually. So I was checking the delegates list earlier, and I saw that there is a quite diverse profile of you all here. So I'm pretty sure that some of the slides for some of you will be trivial, and for some others, might be some interesting aspects. My main objective is twofold, really, for today. One is, for those of you who are not familiar with nuclear medicine / molecular imaging, to give you an overview of what nuclear medicine / molecular imaging is, and from different perspectives, primarily from R&D and what has been the path and what are the main forces. I'll talk a little bit about the market as well. And the next important thing that I want to achieve today is also to give you a fair description of what are the problems that we see with molecular imaging today, and how this impacts on the adoption in the clinic. And I'm pretty sure that some of this will resonate also in in the other imaging areas, but perhaps not as much as with molecular imaging. So one more thing about IPES. So IPES is, probably you don't know about it, it's a trade association of all manufacturers of nuclear medicine equipment that are active in Europe. But also we have the main research reactors which provide the radiopharmaceuticals, and also big companies like Mallinckrodt or Eli Lilly or GE Healthcare that are active in the radiopharmaceutical production distribution within the European Union. And we also have affiliate members which are research institutions, as well as commercial entities which are active outside Europe, but nevertheless are affected or affect the trade and the different aspects of nuclear medicine business in their areas or in Europe. My talk will be about, we'll try to be segmented into three areas. One will be, what is molecular imaging, and what are the enabling technologies. I will leverage from Dimitar's talk on this. The second will be the challenges and the barriers of adoption. I will give a fair bit of chunk of my talk on this, because I think that this is extremely important going forward. And of course, I'll talk a little bit about what is personalized medicine and what the molecular imaging / nuclear medicine role is there. I'm always saying this up until now because, nuclear medicine is basically a construct. We are using words to actually represent what we want to talk about. But together with MRI, for example, up until very recently, or several decades ago, 20 years ago, it was nuclear magnetic resonance. So people dropped the nuclear because of the perception in the population about this with this medical application, and we kind of see the same sort of tendency now also in nuclear medicine. I quite like nuclear medicine, so from now on I will be using more nuclear medicine so that you understand what I'm talking about. What are the potential medical applications for molecular imaging? This is a nice graph that I took from a preclinical publication. You see here that it talks about X-ray, MRI with the different segments of MRI, spectroscopy, and ultra high field like 7 Tesla MR. And then in the y-axis is the molecular sensitivity. So how many information carriers you need to have per volume in order to give you an appropriate signal, while in this area here is the spatial resolution that you are getting out of your modality. So as we go down into the molecular sensitivity, so being more sensitive as a modality, we're starting seeing SPECT coming up, and optical imaging, as well as PET. And you see here that optical is extremely good in picking up very faint signals, but the problem with optical, obviously, of course, is that you have very big attenuation. So it's at this point in time, it's fair to say that it has not been widely utilized or adopted in the clinical domain. So when we're talking about clinical imaging applications, basically we focus on CT and X-ray, of course, planar X-ray, MRI, SPECT, and PET. And we can see things into a different perspective. So if we take this as a reflection of what actually we can study in the human body, we have here, and this is again from a non-clinical publication. So what we are here, what they talk about structural is essentially a structure of tissues, while in the clinic we tend to use structure more about bones and muscles. But nevertheless, you have structure, you go into metabolic, then to study immunological parameters, and then if you're lucky and you have a very sensitive modality, you might be delving into the genetic information. And putting the imaging modalities in this sort of arrangement, you see that traditional CT is up to here. Then you've got MRI, which you can study metabolics, and then you only are left with the nuclear medicine modalities, which are basically in the area of immunology, but PET has been shown that it might be also beneficial in genetics. I personally think that SPECT would also have a role to play here, and I will show you some potentially interesting, almost commercial systems that might help us going towards this direction with SPECT. So nuclear medicine or molecular imaging is by default today a hybrid modality. If you exclude SPECT, that you can buy by itself and you can do SPECT imaging, everything else is basically bundled together with a anatomical or functional modality. So you have SPECT, but you can also have SPECT CT. You can have a PET CT, which is combined by a state-of-the-art today CT scanner, together with a PET ring, which is usually, well, all of the arrangement, the commercial arrangements today is with the CT ring sitting at the back of the CT scanner, of the PET being at the back of the CT scanner. So you have the CT in the front and PET ring at the back. And you have PETAMAR, which is the existing commercial systems, is basically a 3 Tesla MRI system with the PET ring inside the MR bore. So you can have simultaneous acquisition of MR and PET. Having said that, the vast majority of nuclear medicine applications, clinical nuclear medicine applications, are only using the structural information from CT or the functional information from CT potential for localization and attenuation correction. Which means that even if the clinical algorithm prescribes a perfusion CT, let's say for studying the tumor, usually that is an extra CT scan which is done in the radiology department, so in a completely different department in the hospital. And this model changes. There are clinical departments in Europe and in the US which they try to break this model. But it is fair to say that in the majority of nuclear medicine practices or hospital departments, you will be referred for a CT scan with contrast. And if that is inconclusive, then you will get another referral for a PET CT scan. And in that referral, most probably, the CT scan would be utilized, will be used as a low-dose CT, and will be used only for attenuation correction of the PET data or SPECT data, and for localization of the lesions. Now, this is a huge loss of opportunity because you have a system that is able to do full-blown applications on CT. So you have here, these systems have anything from 16 up to 128 slices, and you can have all the applications of the standalone CTs on the PET-CT, and essentially you're using them just for localization. This is not the case on PETAMAR, and this is definitely not the case for PETAMAR. And this is the promise as well as the curse of this modality. So PETAMAR scans today utilize the full-blown sequences that the radiologist will prescribe for MRI acquisition, plus the PET tracer from the nuclear medicine point of view. And I say that this is a blessing and a curse because it actually paves the way towards integrating molecular imaging and actually make use of two very potential imaging modalities. But the curse is because you have two modalities which are fundamentally different from the point of view of interrogating the patient. So one is MR is traditionally organ-based modality, so you will be acquiring scans for your liver or for the brain in oncology setting. While PET is a whole-body imager, you are predominantly interested in metastatic disease. This basically means that you will be spending traditionally anything between 40 to 120 minutes for the MR acquisition. Some of those can be leveraged simultaneously for the acquisition of PET data, but most often you will need extra acquisitions for PET as well. And this drags the scan quite long, and it's not necessarily a problem with the patient, although with oncology, late-stage oncological patients are fragile, so yes, you have concerns about this as well. But the main problem is the economics of the business model, of how many patients you can scan, and the reimbursement model, and I will be talking about this later on. A glimpse of the molecular imaging European market. So this is a model, of course, but up to 2015 is actual data. So this is the blue is the PET-CT. This is sales, and the green is the standalone SPECT-CT, and the red is SPECT. The green is SPECT, standalone SPECT, and the red is SPECT-CT. Now, if I showed you the same graph a decade ago, this thing here would have been reversed. So from the introduction of SPECT-CT, which is around 2006 clinically, well, no, that's multi-slice CT, even earlier, but from 2006 when we have the proper multi-slice CT attached to a SPECT camera, we have seen that most of the departments are now interested in buying a hybrid modality system also for SPECT. Now, if I contrast this one with the applications, it doesn't show it doesn't say the same story. This is, and again, all these are market reports from independent agencies, so take everything with a pinch of salt, but it shows the bigger picture. So if perhaps the accuracy is not to the last decimal point, but it definitely shows it's accurate with respect to what's happening over there. So green is basically PET-CT/PET-MR, and blue is SPECT. Of those, only a fraction is SPECT-CT. So the message here is that although this market segment is diminishing at the moment, the standalone SPECT, still the vast majority of the applications in nuclear medicine department are for SPECT. And this is an imbalance, which we are, in my opinion, we're going to see this growing further, and probably also PET-CT will get some of the applications from this. But definitely the message here is that SPECT it has been seen the last decade as a not so sexy modality, but I personally think that it has a huge room to improve and a very big potential. And it's definitely a very important segment of the market, at least for the manufacturers, as well as, and more importantly, for the radiopharmaceutical manufacturers. Prices. This is, again, a ballpark figure. So I cannot claim that it's 100% accurate. The average lifespan of a system in a European department is anything between 8 to 10 years, perhaps a little bit longer if you have financial constraints, but definitely, and this becomes also a reality in the US now. I think Medicare has already implemented reimbursement according to your image quality performance for your CT scanners, and I think that it's also being implemented for PET scanners as well. So this is the lifespan of a system. You buy a system today, you're going to be stuck with it for the next 8 to 10 years. The average price is around 1.5 million for PET-CT, around 4.5 million for a PETAMAR, and around 400,000 euros for a SPECT system. Sorry, a SPECT-CT system. And the ranges are they have quite a variability as well. Dimitra said about, second one. Okay. So Dimitra said earlier about the transition into quantitative imaging. I mean, this is a huge trend for molecular imaging. And it is fair to claim, and I may make provocative statements. Please don't hold them against me, but hold them in your brain, and we can discuss later on. Quantification is probably the biggest proponent of nuclear medicine. It's fair to say that today it's the only clinical modality that can actually measure megabecs per cc, or information carriers per unit volume, quite accurately. And you can link this information into, for example, when we're talking about neurology, how many receptors have been taken up your tracer. This is all other quantitative imaging modalities are basically, when we talk about quantification, they talk about semi-quantification, so quantification versus a standard. Here we have the ability to go even with SPECT, as I will show you, but definitely with PET, and since the beginning of this modality, you're able to measure exactly how much radioactivity you have in your patient. But why this is important? Why is it becoming more important now than it was before? First of all, because there is an increase in therapy follow-ups. So in oncology. So now you're not just interested to see where about the tumor occurs, but you want to trace this tumor and understand how this, for example, tumor becomes less metabolically active because of your therapeutic regime, and you want to make sense out of your strategy for treating this patient, and you want to know this as quickly as possible, so that to enable you as a clinician to make decisions and take this patient out of a very expensive therapy, for example, because it has a very poor quality of life for him and doesn't make him better, and for example give that same therapy to somebody else with a different genetic profile. Then there is a significant push for for academic institutions primarily, and adopted now more and more by radiopharmaceutical companies, to provide more clinically validated tracers beyond studying of metabolism, which was done traditionally with FDG, which is fluorodeoxyglucose. It's basically an analog of glucose, of sugar. And of course, another point where PET and SPECT quantification was important, was in cardiology, where traditionally in North American market was the main push for nuclear medicine. But also that is with SPECT, but now we see the same sort of push for PET as well, with different traces like rubidium, like ammonia, like where you have access to oxygen. And these applications, three applications together with a fourth one, which is more sort of a trend now, and this is the growth of targeted radiotherapy applications. So this is essentially a targeted drug which is tagged to a radionuclide that emits an alpha particle or a beta particle and basically is linked to the tumor, and it bombards the tumor with radioactivity. And if you are going to inject open source radioactivity into your patient with the hope that you will destroy something, you must be sure that wherever this goes actually is the area that you want to attack and not something else. And you also need to study from a dosimetry perspective how much activity the rest of the body receives. So this is part of what Dimitra introduced earlier, the theranostics approach. So you have the same entity, which you tag it with one tracer, for example, gallium, and you do a dosimetry study, or even feasibility study for the phenotypic profile of the patient. And then once the patient qualifies for the therapy, you inject the same drug with Yttrium-90, for example, and this delivers the lethal dose of radiation to the tumor. Now, enabling technologies for making these four things happen. One is new biomarkers. As I said, there's a big funnel, and it's probably the most active period over at least my short experience of nuclear medicine, that now we have more and more traces coming into the market, and most importantly, we have companies which are committing funds to push this from phase 0 to phase 4. We have new detectors, new reconstruction software, new post-processing software, so new technology which enables the better image quality and more accurate quantification of the data. And we have technology which has been in the past and has been used by academic institutions, but now more and more we see that transitioning into mainstream clinic through tailored applications from either same vendors that they sell the systems or from third-party software developers. So what are the enabled technologies for molecular imaging? The first and foremost is radioisotopes, radiopharmaceuticals. And I say first and foremost because if you, in contrast with, for example, MRI or X-ray, where you excite the tissue or you irradiate the tissue, here you must have a radiopharmaceutical that you inject to the patient or you let the patient inhale in some occasions, and then you do a measurement. So there is facilities where they produce the radiopharmaceuticals, and these radiopharmaceuticals are basically transitioning or moved to quite fast because of their short half-life into the hospital where the imaging happens. But also we see more and more hospitals are buying their own cyclotrons for PET primarily imaging or predominantly imaging, and then you have the ability not just to do fluorine-labeled radiopharmaceuticals, but also other applications, for example, perfusion imaging with oxygen, or carbon-11, which is more used in research, or nitrogen, which is predominantly used for cardiac scans in the form of ammonia. But also, we have generators. So apart from this is the simplified supply chain for a generator. So let's assume that we're talking about SPECT, so technetium. You have uranium targets in the research reactor, which are irradiated. Then these now radioactive materials are moving to a processing facility, and then which are processed into a purified, and then this goes into a generator manufacturer facility. This can be quite far away from each other, so all this means that there is a transportation and legal aspects and regulatory aspects, particularly in our era, where there is a huge concern about security, and where potentially these areas, these products find the right owner, or potentially end up somewhere that they shouldn't be. And there is a significant effort from the regulatory and legal aspects to harmonize and simplify all this. I remember, for example, not so many years ago, there was a problem with a massive airplane company here in Europe, where they decided that it was too much to handle radioactive substances in commercial flights, so they will stop carrying the radiopharmaceuticals. But they had some flights which they were the only company that were reaching some areas, and that meant that the whole business model or the whole patient population in those areas wouldn't have access to technetium. So again, that was when entities like IPES come into play and start to negotiate what and support these companies to actually facilitate the transportation of the materials. The cyclotrons themselves, which are huge entities, big systems, we see the last years that are becoming smaller and smaller. So there are two companies at the moment. This is the system from ABT, but also GE Healthcare has another similar size cyclotron, which you can have it into your department. You don't need to build a bunker around it. You basically don't need a huge radiopharmaceutical team. You just need a automatic dispenser of some sort and a much smaller radiopharmaceuticals team. Just to give you a description of what kind of numbers taken from the marketing materials of this company. So you see reducing your footprint of your facility. You see reducing the structural requirements of your facility, which means that you might be able now to put it into an already existing space rather than develop your own specialized facility. The cost of personnel to handle this is becoming smaller. The operational expenses become as well smaller, as you would imagine, and also the product cycle is smaller. What doesn't say here is, of course, that you are now have to press the button, take one dose, then wait, then press the button again, take another dose, while with the full-blown cyclotron, basically you start the production in the morning or during the evening, and you get your doses, and you can have your batches for what you need during the day, and perhaps if you have clients, you can sell the extra doses to other hospitals. So these are completely different business models. But what I want to say here is that now you have the ability to choose which model fits you best if you want to have PET images. And these sort of modalities allowed, for example, several hospitals in the Maghreb region, so that's the North Sahara region, to now start experimenting with PET-CT in their departments and starting to offer this sort of imaging modality also in their departments. The radiopharmaceutical production in the hospital. When I started not so long ago, I think it was like 20 years ago, the radiopharmacy department where you would be creating your radiopharmaceuticals looked like this. And if you had a crazy scientist, because that's what they were at that point, and they wanted something else rather than what you were preparing, then you had to scrap all this or put it aside into another cubicle. Everything here are lead bricks. I still remember Royal Marsden having a large variety of bricks for radiopharmacists to use. And you would have to build another structure like that in order to produce, for example, a different radiopharmaceutical. Now, we have moved away from that. So now there are radiopharmaceutical companies that they produce automated dispensers. So this whole thing has been transitioned into a cassette. If you want FDG, you put the FDG cassette, you press the button, you do the dosimetry, they produce the report, everything is GMP, and you have the tracer that you can inject into your patient. And what at least I saw in one of the Nature journals, and I was quite impressed, and I think that this is the way forward, is Microfluidics finding their way also into the radiopharmacy department. And this is a huge news if this becomes reality also in the clinic, because that means reduction of cost. And in general, this transition from here to there means that first you have a very good way and simple way to have GMP compliance, which is important at the moment for getting a license to operate. You have a tremendous cost reduction. You have reduction of waste. You have simplified way of producing radiopharmaceuticals, and more importantly, you are eliminating the human error, which inevitably it's there. So this is another enabling technology or enabling stream of technologies that made molecular imaging of today actually exist. So going into a more familiar, perhaps, space, I want to give a very quick overview of the innovation in PET. PET is a relatively new modality. It started in 1953 as a concept. This is the first image from Brown and Swindle, which is basically a patient with a tumor, and they were using two planar detector and a printer to see where the density of the photons that the detectors have been detected. And then from I think the same department, they created this first prototype, the positron camera, the MGH positron camera. And then they created a company, and they, in around 1970, early 70s, you were able to order this system and use it for in your department. Around 1970, end of 70s, we have the ring-based PET scanners, which is, I think this is important because this is the technology that basically established itself, and all of our PET systems at the moment has this geometry. In '82, believe it or not, we have this first time-of-flight system. The problem at that point was that the technology was not ready, and also the applications, which were predominantly neuro applications, were not appropriate for the timing resolutions that this system was capable of achieving. So time-of-flight PET just became an oddity and was just or a wishful think, if you wish. And then at some point there, FDG was developed, which is the reason why PET exists at the moment in the clinic. This is the radiopharmaceutical that you inject, and you study the metabolic, how fast the cells consume glucose, which is one of the main fuels of body, and with the hypothesis that cancer cells are more active, therefore they utilize more more glucose. The fluorine here is a non-reversible kinetic model, so the fluorine that is trapped into the cell, it remains in the cell until it disintegrates. And therefore, you leave the patient to uptake the medicine for about an hour, and then you start imaging, and then you see the lesions having accumulated the fluorine. This gave rise to a variety of commercial entities, which now they had products that clinicians can take and use it for their patients. Gamma cameras picked up, and you could also have coincidence imaging with a gamma camera. Image quality was not that great, but in any case was the cheapest relative, and you could still visualize the disease. Many hospitals went for this approach. And then in 1998, you have another breakthrough, and it's the combination of PET with CT. And I say this is a breakthrough because it made two major things. One is reduce the amount of time that you need for a scan to a fraction. And this is because before, in order to acquire the attenuation correction, you had a source rotating around the body, and you would acquire this data and you would reconstruct the attenuation map. But this was taking forever. So now by having the CT, it was just in a snapshot that you would get the CT image and you will use it for attenuation correction. So that was a workflow advantage. But also, and most important, was a PR advantage, because all of a sudden, before you had blobs in an empty space that you had as a nuclear medicine physician to say, oh, all right, so this is the body, this is around here, so this is a lymph node. Now, and you had to justify this to the referring physician, and you have to argue with the radiologist. Now, you have an overlaying image of the anatomical map of the body, together with where the lesion exists, shown by PET. The first commercial PET-CT scanner was by GE, and was the Discovery LS. I still remember me being a student and going to the old UCL hospital to see this marvel. Was the first system in the UK. In 2006, Philips introduces the first time-of-flight PET system. This happened because at this point in time, you had the right materials, so the right scintillating materials, and the right electronics to actually back this kind of approach. Timing resolution was about 600 picoseconds. That means that it was quite a good system for whole body imaging. You would see better contrast, for example, in the abdomens and in the lungs, but was basically no point to use time of flight for the brain. In 2008, I think it's another breakthrough, and this is the introduction of solid-state detectors. So Siemens introduced the first simultaneous PETAMAR system for the brain. That was an insert that you would pull into a 3 Tesla MR scanner, and you would actually simultaneously study the brain from PET and MRI. In 2010, there is the first clinical whole-body PETAMAR system, from Philips. But as you can see here, this is the same time-of-flight system that you had there, but the MR is quite far away, so this is not simultaneous. You had to put the patient on it, you had to go first into the MR, do the attenuation correction, rotate it, go do the PET acquisition, then rotate and go and do the MR again. Doable, but not the best approach, and that's the reason why it didn't last, unfortunately. I was part of the team which developed the system. So in 2011, actually it was almost beginning of 2011, Siemens came up with the whole body integrated PETAMAR system, which was based on avalanche photodiode detectors, and it's still a product. Since 6 years, they have hold this whole system up with significant functionality about motion correction or motion compensation in PET with MR, better acquisition, etc. In 2012, Philips introduces the first prototype for time-of-flight PET-CT. So this was essentially the same system as this one here, only when you opened the gantries, you would pull in the detector ring, this SIPM detector ring, and you would image the patient. And in 2016, GE Healthcare essentially introduced the first commercial silicon photomultiplier-based time-of-flight PET-CT system. So this is a trajectory of where we started, and I'm sure that I have missed things around here, but I think that it's quite the most important points are there. Now, moving into SPECT, I utilized a very precise list from Professor Brian Hutton from UCL to demonstrate the main points of the technology innovation. So in 1958, we have the first gamma camera, or the invention of gamma camera by Hal Langer. In 2063, you have the first SPECT system described by Kuhl and Edwards. Late 60s, beginning of 80s, we see at least 3 groups trying to do SPECT with rotating the chair. This is the Japanese group in the picture. And then we've got the first SPECT using a rotating gamma camera in late 70s. We've got in around 1983, the commercial SPECT is introduced. In 1987, we have a patent from USSR about SPECT-CT, although I don't know whether there is an actual system, but definitely the first SPECT-CT system as we know it in the West has been introduced by GE in 1999, and that was the Infinia Hawkeye, actually the Millennium Hawkeye, I think was called. And then you have 2 more important points. One, you've got the rise of iterative reconstruction algorithm, which revolutionized the tomographic reconstruction space. And you have the introduction of the first multi-slice-based SPECT-CT by Philips, which was essentially a brilliant CT scanner, 616 CT scanner with a skylight attached to it. In SPECT, I said that there's substantial room for improvement. It's the same with PET, but in SPECT in particular, because the space was made static for quite a long period of time. And I want to bring into your attention 4 products which are either works in progress or now in the field and are quite interesting steps forward. First of all is this from a small company called Molecular Dynamics. It's a CZT-based high-resolution brain scanner. I've seen a couple of images and are quite impressive. And almost the same image quality is achieved by another small company's system, which are predominantly active in the preclinical arena, but now they are also they're introducing the new system in clinical. And this is a standard technology with multi-pinhole collimators that they utilize magnification to improve spatial resolution. And the images are quite good, quite impressive. Last year, I think, or definitely this year, GE Healthcare introduced this year, commercially, the first whole-body CZT-based SPECT-CT, which actually the business model is also very interesting because you can have your NM670 traditional system, and then you can just order the new gamma camera, and they will unplug the heads and plug in the CZT heads, in your hospital. And another system which I put over here for completely different reasons is the Siemens Symbian Intuivo SPECT. And the reason why I put this system here is because it's the first system that has FDA claims that can do quantitative imaging. So this system first utilizes the anatomical information from the CT to correct for partial volume effects in SPECT, and also it calibrates the system. The system is calibrated so that it gives you, for 4 radiopharmaceuticals or isotopes, quantitative information. So, in my opinion, these 4 systems here sort of show the direction where SPECT is going. PET-MR. This is an area where it's very close to my heart. Not so long ago, you had the ability to choose between 5 different settings. Now you have the ability to choose between only 2 systems. Both of them are 3 Tesla integrated. You can do PET simultaneously with MR. The enabler, the technology enablers that for these systems were basically the solid-state detectors, the multi-transmit, receive, or other technologies that speed MRI acquisition, improvements in PET sensitivity, improvements in corrections of PET. That's very important. Still work in progress, but tremendous steps have been made. And of course, novel MRI sequences, which have the ability to produce the attenuation information for PET, quantitative PET, and also the ability to, I dread to say this, but potentially allow PET to go beyond FDG by doing metastatic disease analysis with, for example, whole body diffusion weighted imaging, and utilize more specialized tracers for interrogating, cancerous receptors or whatever you think it's, or proliferation or some other parameters of cancer. It's a very complicated modality, but it has the ability to do magnificent stuff. And what Dimitra was saying earlier about the combination of modalities, I think actually, Dimitra, that we have it already in the clinic. It's not very widespread. I think there are currently 140, 120 systems in the globe, but you are able to utilize all the parametric information from the MR together with targeted information or fully quantitative information from PET in either dynamic acquisition mode, or utilize MR information to correct your PET data, or utilize PET information to actually make sense of your MR data in a more quantitative way. So this is the stuff that I wanted to get rid of, and this is the stuff that I want to focus on. And I think I have only 10 minutes, which is challenging, yeah. So first of all, and I'll go quite fast through my first slides, there are significant factors which happen at the moment in the global healthcare system and affect the all imaging modalities, and nuclear medicine in particular, because it's a niche modality. These are segmented into 4 categories. We can argue about there are different segmentation, but one is about the demographics. So global population increases, quality of life improves. There are average increase of age of patients, which gives rise to other facts like increased number of patients per department, expectations of the patients for how to be treated, the prolonged duration of chronic illnesses. And then you have economic factors, and we have lived these very vividly the last 10 years. You have globalization economy, which synchronizes economies in different parts of the world. So if one world has a problem, all of us suffer. The reduction of commercial barriers is another very important aspect, which essentially increases the competition in all areas, and that is competition in the manufacturer space, but also competitions in health systems, because now, if you want to have better treatment and you don't think that you will receive it from NHS, if you have the funds, you can go and do that somewhere else in Europe, or even Singapore, for example, or in the US. you've got novel technologies and you have easier access to information. This improves the outcomes, of course, the novel technologies, and you have better educated patients which demand appropriate or what they think that it's appropriate for their case. And you have, of course, the cultural assimilation, which first of all, it makes, and this is quite right, obviously, it makes patients in one part of the world to have exactly the same standards and demand the same sort of therapies for them, for their illness, as, for example, in other parts of the world. And you have, quite importantly, globalization of diseases. So, for example, diseases which were predominantly like stomach cancer in Southeast Asia, and they were not that predominant in other parts of the world for the last 40 years. It pop up more and more, for example, in California, where you have a big Asian population. So all these macroeconomic factors affect how the health systems react, and how R&D is successful in bringing new technologies into the field. This is a very complicated, it seems, graph, but it's not actually so complicated. It basically shows the four segments of developing technology. And you have here the innovation phase, which is the research. But you have also a second innovation phase, which is becomes more important, particularly for disruptive innovation like PETAMAR. And this is about figuring out how to utilize this new technology in the field. And you have the players here about affecting these stages. And this is a prelude to my next slides. You have bottlenecks here in regulatory bodies. You have problems in the perception of the referring physicians for new technologies, right? I'm talking about new technologies. And you have a very big bottleneck on the payer side on how reimbursement systems actually define the reimbursement level for a new technology. If I want to focus on molecular imaging, there are things that have happened, as I said about in the beginning about quantification, but more in general about nuclear medicine, that actually have boosted the field. And these are, for example, the proof that there is significant improvement when you are staging a patient with FTG, for example, for lung cancer or for non-Hodgkin's lymphoma. There is the combination, as I said, of PET-NCT and SPECT-NCT gave a boost both from a productivity point of view as well as from interpretation point of view. You have novel indications that are coming into the field, and believe it or not, even Germany, which is the most conservative sort of health system with respect to adoption of PET, actually now started to be motivated and think about potential advantages of this technology. You have better access to tracers. So you have now, as I said, generators. You have, and the chemistry that goes behind it. You have novel business models, and you have the new, smaller cyclotrons that enable you to do stuff in areas where traditionally there wasn't any option of having a PET-CT scanner. Reduction of costs is another driver. There is a clear focus of the manufacturers at the moment on the value base. So all of us, and when I say all of us, I mean the 3/4 big manufacturers. We have our flagship systems. But if you went to the Nuclear Medicine Society meeting in Barcelona this year, you would see on the floor of 2 out of 3 big companies to introduce value segments as well. You have radiopharmaceuticals, which are prices dropping. Is it this good? It depends. We can discuss that over coffee if you want. And finally, you have new manufacturers which are either entering or re-entering the arena. And this might be a problem for established manufacturers, but it's definitely good for the development of the modality. It shows activity. There are other potential drivers. So amyloid imaging is one. We hoped for Eli Lilly to get the FDA clearance for their anti-amyloid drug for Alzheimer's. Unfortunately, that didn't happen. There are other amyloid drugs, dementia drugs in the pipeline. If this happens, and if, as everybody thinks, the first dementia drugs to be based on the amyloid cascade hypothesis, amyloid imaging, which are now by 3 manufacturers, that's GE, Piramal, and Eli Lilly, will be able to have a boost in their sales, and that means A, that new drugs will be developed, B, that there is a positive feedback, amplification feedback on these companies and other companies to actually be more active in pursuing regulatory approval of their tracers. The pharma companies more and more seem to start embracing molecular imaging. There was a push around 12 years ago where everybody was setting up their own imaging centers. This model has failed. But now there is significant push from pharma companies to utilize external agents and use not just MRI and CT, which is predominantly the modalities of choice, but also PET and SPECT for certifying their clinical trials. And of course, is the rise of theranostics, and here I have only AAA's Lutathera, which is a tracer which has just been introduced in the US and in Europe, and it's basically a lutetium-based theranostic tracer for gallium lutetium for a lymph node metastasis. Sorry for the problems with market acceptance is basically outlined here. As we go from the 80s into 2000, you have increased demands of outcomes mainly, and increased demands of demonstrating cost effectiveness. These are, in my opinion, the most important problems. A very quick example of the outcomes. Regulatory authorities, to give you approval of a new tracer for a diagnostic tracer, irrespective of whether there is imaging or anything else, you need to demonstrate not that it works, not that it detects what it says it detects, but that it affects the outcome of the patient through the therapy that the doctor will prescribe based on the improved information from your diagnostic. Very simple example, if you have an increased diagnostic accuracy of 30%, and you have treatment changes in 50% of the patients that you have tested your diagnostic, and you can demonstrate 30% improved outcomes in that patient cohort, then you end up having 4.5% of improved outcomes that you can go to FDA and argue about it. Now, this basically means that you need to have massive clinical trials for a specific indication that will signify boost your cost up. And trust me, radiopharmaceutical business is not as similar to pharmaceutical business with respect to revenues. Cost effectiveness, I don't have the time to go through this. What I would say is that you need to demonstrate that your new technology is better than the previous gold standard to basically be reimbursed and replace an older technology. The problem with molecular imaging, and probably with all of our imaging modalities, is that the technology changes very rapidly. So you have every 5 years, you have new technology out there, either it's detector or reconstructor. That means that you have very short window to pursue and actually capitalize from your investment. You have a very small patient population because by default, this is what molecular personalized medicine is all about. If you are going to produce a product for personalized medicine space, and you're targeting for thousands of samples, then good luck. people try to overcome this by utilizing modeling. The credibility of models are not so much accepted in the traditional HTA's process. So it's timely and cost. And I will quote here a friend from GE, Dr. David Lee, who is the general manager of market access in the US. And he says that perhaps in this particular space, it's good practice to have the evidence. But if you're waiting for cost-effectiveness evidence, actually at the end of the day might not be cost-effective itself. There are, finally, my take on the areas of concern, and I will finish with this slide. One is the market access for new radiopharmaceuticals. In PET, in SPECT, it's not like MRI. You cannot test a new pulse sequence, and it's non-ionizing radiation. Even in MRI, in this aspect, is quite difficult. Don't get me wrong on that. But you have to have a radiopharmaceutical. And this radiopharmaceutical, in contrast with the pharmaceutical industry, has much lower sales volumes and much lower revenue. So the market access is of paramount importance to be much faster as it is today. Reimbursement is important as well because, for example, as I showed you, the majority of scans is SPECT. This is nuclear medicine. This is technetium. Now, the technetium is not adequately reimbursed in 25 out of 28 EU studies. Now, if you now factor in that we need to change from high-enriched uranium into low-enriched uranium targets and factor this into the business model, this is a disaster. There is lag in patient referral, so there needs to be education of physicians about new technologies. And of course, is the current economic climate, which we can't do anything about it, rather than think of more efficient business models. And I want to thank you about your attendance.